I write so slow, it's exciting to do runs. I did one Monday and the other one.
I don't want to get something nasty.
Alright. That's 52.
No, that's what it did the other day. A lot of these are like really strong. What I've been doing is making something and then
Good. What are you guys doing? Good. Is this everybody's work?
Yes. Are we gonna get the first one back? First,
You
second,
can if you come to my office. Thank you for those of you guys who did this. Although,
Thanks for the opportunity.
Yes.
you're welcome. So hopefully this will help you guys out. Did you guys find this a useful exercise, a tool? What was the best part about it? What was something that worked?
Realizing I chose the wrong answer when I was down to the right answer. So like realizing like I had the right answer, but I chose the wrong answer.
Yeah.
That makes sense.
Second guessing.
So I was down to the 2 right answers, but I chose the wrong one instead of the better. I can't explain myself. I'm sorry.
It makes sense though, but yeah, I saw that a lot.
Okay, so basically it helped you to kind of reflect on the
Like, I
decision-making
knew
process.
it, but I overthought it.
Okay, yeah, so that can be a pretty useful thing. That's one of the reasons I think this is a helpful exercise, because then you can figure out where did I go wrong, how did, not where, but how, right? You can start to think a little bit deeper about, because I know you guys know the stuff. We just got to get that to translate to the test, which is just these little details, right? So
That's
hopefully
the
this.
way some of the questions are presented too. It kind of makes you go, what? Like, yeah, just always present sometimes. It can you off.
Yeah, so that's why I try to give those little tips of how to manage the way that the questions are presented, which is, you know, work through it slow. Just figure out what is the one thing it's asking, and then let's just find the one answer that best, you know what I'm saying, and kind of simplify so overthinking it and stuff like that. What did you guys find any other usefulness to the exercise that it was helpful with?
Looking through the notes again and the book also.
So going through the notes again in the book. Okay, anything else?
Why exactly you were wrong?
Why? Yeah, so not just sort of thinking about, you know, knowledge as regurgitation of semantic facts, of just here's the thing, but now starting to kind of reflect a little bit deeper about why. Yeah? you guys find that experience? It make you go back and like double check stuff and figure out like, especially the ones you really thought were right. Yeah.
It helped me learn that I kind of chunk similar things together. Like on this last exam, I mixed up substance P and is it
Nociceptors, yeah.
Nociceptors. Like I mixed a lot of those up, and then some of the sensory matter. Like I knew that something was up the dorsal column, but I mixed up the pain pathway and the sensory motor, and I go, oh, okay. But I knew. I just kind of put everything together.
Yeah.
I did the same thing with like acetylcholine and
Totally.
and substance P, so it helped me
Yeah,
parse things out.
it's a common thing. You know, it's easy to confuse stuff because especially the stuff you kind of know that you've been studying. But I hope that the process kind of gets that reflection, thinking a little bit more about, you know, that transition that people talk about when we start to study and when we start to try to learn things, not just for the information. Like, we know that substance P is definitely related to nociceptors because there's a link between them, right? But it's not because they work on the nociceptors. They're just both linked to the topic of pain and the sensation of pain, but they work in the understanding of the knowledge is different than just recognizing those things, right? So a lot of times, you know, in sort of, you know, K through 12 education that we're all kind of raised through now, we're kind of, you know, taught to tap into just identify these facts, but don't relate them together as much. So hopefully this will help to kind of relate that stuff together. Did you guys find that? Yeah. Okay, cool. What about the first test stuff? Did you guys go through and do that one too? What do you think about that now looking back?
Because you knew all the answers, like, duh, yeah, like.
I think I should have seen you during all of that.
I also think that.
Well, like I put for like the gas for nitric oxide, I put gaseous.
I did the same
And it
thing
was gas. I'm like,
instead of
oh.
Yeah. What else struck you about the first test? Any observations about it from the back? You can hear a lot from all you people, very vocal in the front. Yes.
I think the exercise in itself is just like it kind of forces us to break down the question. Because you know, when taking it, we're kind of going through it, and we're giving time and just trying to get out of here. But I think having to go through what we got wrong, the reason why we got wrong, and then why the right answer is right, it kind of forces us to be like, okay, I was just stupid. So.
Well, I didn't say that, but.
I mean, it's just kind of like it makes us think like, okay, if we would have took our time in some cases, we probably would have got more right than wrong.
Yeah, totally. And you know, I totally agree with you, man. I have the same experience when I take tests and all that stuff too. And it's funny because we have, you know, 2 hours. And we get it done in like 25 minutes because we're still trying to rush. But you're totally right. I think that it with a little bit of reflection, it kind of taps us into those ideas of that. And hopefully we can take that forward though, right? Not just looking at what did I get wrong and why was the right answer right instead, but also, like you said, to do the exercise, you had to really dig into the question and say, well, what is it asking, and why was these answers the right way to think about it? Sometimes on really subtle facets, but really slowing down and going through it. But wouldn't it be a whole lot easier if we approached the final that way beforehand rather than afterwards? You guys follow what I'm saying? So that's why I keep using this exercise to practice with, is because hopefully we'll now hopefully you guys are experts in looking at the questions in this way now and thinking about it like a little bit slowly and going, oh yeah. Let's just take this one thing at a time. Take that, adopt that. If you adopt that strategy, we can do that at the test too, right? You guys got one more test left, and hopefully you guys can see a lot of really, really, really good opportunities that are still left for you to do really well in the class. You guys see that? Right? If you do the math, think about the points you get back from our extra credit projects. Think about being able to do the math and you know, work your little algebra equation out. And there's a lot of really good opportunities that a lot of people have to do really well. Yes, sir.
I will say I do wish you would consider letting us write with paper, because like one thing for me, I'm super tactile, so usually on exams I'll cross out the right and wrong answers, and it helps me think about the question, like on the question going up the dorsal column system. When I said it a lot, I said, oh my god, no, that's ipsilateral. And I chose contralateral, and I knew it possibly did that, and I just confused myself. But when I'm so used to physically writing exams, it helps me like parse that out.
I totally hear you, but we have a really practical limitation there, which is that you don't have desk space to write. You singularly do, because you have a very large desk. But a lot of everybody else has a tiny little desktop that's already occupied by a laptop. So, there's always alternatives for that on campus if there's resources to pursue if you need some of those. So we can talk about that if you want to after class, but for the most part, I think it's a good exercise to be able to think it through, like Donovan was saying earlier.
Well, that we also learn it because we have to learn it to answer the question. Isn't that the point of the class, is to learn this?
Yes, it should be here.
So, I mean, you got the wrong answer, then you learn the right answer, then you learn.
Mm-hmm.
So it's taking place.
So Donovan, the thing you mentioned earlier too about answering the questions and thinking through, I've always thought of it as kind of like being like a witness on a stand in a court case, right? Right? You have to slow down because they asked you a question. And it's kind of an important question, and everyone on the jury is waiting to hear your response, right? So you want to think really carefully and deliberatively and just say what you need to say. Is that kind of how you experienced looking back on these questions now?
Yeah, I mean, looking on the first exercise when we did this, it was just having to do it and knowing that I had to get it done because I mean everybody here wants to get their points back. So you get it, like I said, it kind of forces you to, okay, now I got to put it to work into actually look for the reason why the choices were made.
Totally. And my message to you is to take that lesson forward to all your classes in the future. To all your jobs in the future, every memo and business meeting and whatever forward, do all that stuff beforehand rather than having to chase it from behind to get the points back. It's a really, really useful strategy, but it's hard work to learn and do. And I mean, I didn't learn it until deep into grad school and a lot of crying on my part. So, yeah, I mean, I think it's a really useful tool to approach that stuff with. Did you guys look back on the first test and find that I might have been honest with you in telling you that it was the easiest test you'll have all quarter?
No.
You didn't think it was? No?
That was the hardest one.
You thought the first one was hard?
For me,
Yeah.
but that's just a very subjective personal thing.
Okay.
Well, usually, like for me, it's usually everybody. For me, every instructor kind of has a slightly varied style. So once I saw kind of your style and the way you ask things, it kind of helped me prep the other one.
Cool. Okay, well, that sounds good. Yes, ma'am.
So the final, is it droppable?
Is the final droppable?
Yeah.
Like one of the
Since
lowest
you dropped
grades.
one,
Yeah,
so
like
yeah.
as far as if it's the lowest grade.
As far as I'm aware,
Okay.
but
I just wanted to-
I'm darn well hoping it ain't your lowest grade.
No.
After all, we all just been through the last 8 weeks. That better not be your lowest grade. We've all been working hard to make sure that's not the case.
I just wanted to make sure we're on.
Yes.
Yeah, so if you're happy with your grade in the class, can you consider it safe then, if you don't do as well on the final?
I mean, I think that's basically the same question.
Yeah.
But
You said
Yeah, but she asked it this time versus her. A different person asked it.
so I think, indulge me if you might for just a moment. I think what you're asking is, do I need to show up and put my butt in the seat and take the final?
No, of course I'm gonna be here, I'm just wondering.
Yeah, because the answer is yes.
I know you
should
have to take it, I'm
Yes.
just.
Yeah, it's
I'm
not
not trying
gonna.
to be lazy or get out of anything or anything like that, if I'm
Okay.
studying all that, I'm just wondering.
No, it's a
can
practical
can
question. It's
Can
a
I
practical-
relax a little bit
It's a
in my stress levels?
Yeah, but yes, at a mathematical level, yes. If that's your lowest score, then we already know since the first day of class that your lowest score gets dropped. So the math hasn't changed, right? That's just raw math. However, what I would encourage you with is not to, wherever you're at, don't be happy with it. Strive for more. You can do better. Why not shoot it higher?
Yeah, I know.
So
I'm
don't
not saying I'm
throttle
not,
back.
I'm just,
Full throttle it straight ahead.
we want to protect what we have.
It's already protected. I've already protected it for you. It's already protected. You can drop your lows.
Okay.
So you can't go anywhere but up. Mathematically, right? Yeah, word. I've already protected it for you, Sarah. So dial in.
Don't worry, keep happy. You can do this.
Yes. Dial in. You get like this is the last chunk of the quarter for people to do really well. Yes, sir.
Do we get our things back before the exams?
Yeah, it's a study method.
To help us study?
Yeah.
When Jim asked that last time, he asked Jim if he had an attachment to the paper. I remember that because I thought it was really funny.
That was the
Well,
first time.
What?
I'm not. So. Yeah, I mean, yes, you can get your papers back if you want, but if you want to study the questions again, just do the assignment again.
That's good.
I mean,
Actually, that's good.
yeah.
Repetition, yeah.
No, seriously. I mean, I'm not saying like I'm gonna hold I need some time to, with them. Like, we gotta get to know each other a little bit. Like
You gotta fondle them a little bit, and finger through them, and came out with
we're just kind of flirting right now. We're just kind of looking and getting to know each other. But yeah, we get them back. Just give me a little bit of time, and we'll work on that. But yeah, I mean, don't think that you can't do that again. It's a good strategy for all your classes. Like it's a good thing to practice with. So anyways, we got that. There was a lot of really good improvement on the test. You guys did a great job overall. Okay, hopefully you guys, obviously you know how you did. So most of you guys know that and can hopefully come away very encouraged from having seen your hard work start to pay off, yeah? I've heard a lot of people tell me sort of independently that they can look back now on that first third of the quarter and realize, wow, I could do a lot more than I thought I was capable of. And so my hope is that when we take this final, next week I think, right? Next Tuesday.
Wow.
Yeah, word. That I'm hoping to see really good scores.
You will.
Because I think it's going to reflect how much you guys are working, how hard you're doing, and really pulling things together. Yeah.
Is it going to be the same amount of questions?
Probably not.
Wait, what?
It'll probably be more than 50 questions. It might not be. It might be 100.
Yay!
It might be 51. It might be 51. Okay? The amount of questions doesn't matter. What matters is what you know up here, and you guys are on track. You're doing good. So practice those active learning strategies. There's a lot of these opportunities to finish strong and do good. Create your own study guides. If you don't want to do the assignment to redo it, create study guides. Come up with tests to test each other. Come up with quizzes to quiz each other. Work those online quizzes, and practice working and interacting with the material. That's what the project like this was designed to help do. So, so there's that there. Were there any specific questions on the test before we kind of stopped talking about the test? Were you guys able to resolve most of that through the project? Yes, sir?
For example, on example, and I don't know if I'm too late on the draw, if some of us did put like gaseous instead of gas, could we get those full 2 points back for the nitric oxide?
Technically, wasn't that the point of this doing?
You're talking about the first test? No, we're not doing with points on the first test right now. You guys just got a bunch of points back on the first test, and we already resolved all that stuff. Sorry. We will that test behind a long time ago. I was talking more about the one we just took last class. So hopefully we're good then there, and hopefully you guys have a breath of fresh air of encouragement, right? You guys' grades are shaping up, yeah? Okay, cool. I told you that it would not be easy, but it would be worth it, and I'm hoping that you guys will come away with that experience. Yeah, perseverance, exactly. You know, I want you to hopefully walk away from the class knowing that you really learned something authentically, and that you earned what you learned. And hopefully you guys are experiencing that, and I hope that we continue towards that. Sound good? Okay, so don't forget today, after class, what are you guys gonna do?
I'm gonna pie your face.
Yeah, you're gonna throw a pie in my mug? Okay, cool. So have that charity fundraiser downstairs outside. So that's there 3 to 5. I'll be there taking all of your results. So send them.
And they have sprinkles.
Oh, all right, they have sprinkles. That's fantastic. Okay, so we'll do that. We're going to talk about learning and memory today. We'll pick up from where we left off. Let's see here. Okay. Just as, I guess, a final note as we hit this home stretch, as I know you guys are doing really well now. You guys feel like you're on track now? Yeah? Yeah, I kind of dialed in a couple things. Cool. So, I got a note from a student yesterday. I thought I would share it with you. And it said 1, 2, 3, 4, 5, 6, 7 points. It said, there's no name on it, so I don't know who wrote it. But yeah, I found it yesterday.
Where?
It was in my office. It said, A, I'm going to fail miserably. It does not reflect lack of effort. I have been trying, and to a certain extent am interested and want to learn this. I'm not dumb, and it makes me feel such. I have meant to get help, but tend to miss catching you in the office. I'm very independent, and try to force myself to learn it, but just don't have enough background to rely on just me. I am trying, and I do want to learn this and do well, but it gets to the point where it overwhelms me and saturates. Can you guys identify? You know the sentiment?
Yeah.
You know the sentiment? You can identify? Yeah? Do you guys know the student? Anybody?
No.
It was me. I found this. This was a note I wrote to my advisor my first year in grad school. Okay? I found it in a bunch of my old files.
I can't imagine you writing.
I know, that's so cute.
You're such a self-assured, like, confident person.
But it's
that
okay
you
to
do
do
it.
sometimes.
So I cannot picture you writing that.
Well, I did. You can match my handwriting later. But I wanted to share it with you guys so that you know that I know the feeling. I have been there. It is not an abnormal feeling. It is a normal part of the process. But it's just the start of the process. It doesn't mean it's the end of the process where it's an excuse to be like, oh, no, it's not for me. I'm not going to stop.
Yeah,
No.
failure's not fatal.
No, hell no. So, those are just beginning nuggets, and it's at the start of the journey. And there's, hopefully you guys have overcome that already and are starting to do very well and excel and know that that feeling is not something unique to anyone, but we all have it, and we can push through and do really well. Okay? So hopefully that can help take solace as you all start to excel in all your finals and finishing up the quarter and all that. Okay? So, without further ado, would you like to pick up where we left off with Hebbian learning?
Yes, yes.
Yeah? Okay, let's dial into...
Be on the final. Let's start it.
Well,
You skipped the first part of the chapter, didn't you?
Yeah.
Like,
Part of it, you skipped through it, said you would come back.
the stuff with the patients?
Yeah.
Yeah, we'll get to that.
and like declarative and non-declarative.
Yeah, we'll get to declarative and non-declarative. Those are near and dear to my heart.
Okay.
Okay, let's start off, and we will go to where were we? Hebbian learning. Okay. So
Weren't we at long-term potentiation?
we were at long-term potentiation, and that's a very direct connection with Hebbian learning. So I know we had. All right. So, Donald Hebbian, if you had to summarize what we said his rule was last class, what would you summarize it as? Do you remember?
Cells that wire together, fire together.
Cells that wire together, fire together. Or cells that fire together, wire together. And cells that are out of sync lose their link. So you talked a lot about this class about cell firing, action potentials, neural communication, electrochemical signaling. Okay? Cells communicate with each other. You guys are experts on this now, right? Okay? But when they communicate, it induces these synaptic level changes in plasticity, structurally and chemically, that we introduced last class. Right? We talked about how you can have synapses grow. They can grow extra synapses. They can grow bigger connections. Right? But then we also talked about synapses retracting. Right? The Taylor Swift synapse. Because it's never, ever, ever getting back together. Okay? With this other synaptic partner. So we've seen these things that can change, okay? And so signaling and the firing is what's going to do that, okay? And we'll talk about that in the model that comes about from our understanding of it called long-term potentiation, particularly this idea from Hebb that neurons that fire together wire together, and of course, those that are out of sync lose their link. Okay, so this ties together other ideas that we've introduced in class in terms of our systems level processing, which is how is information being transmitted in a lot of our sensory systems? Well, through rate codes, remember? The rate. So how often, how frequent that information is coming in, right? It's a really fast train, bop, 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 versus slow. Pattern of effects, right? So that will change whether or not those firing patterns will change the actual structural connections between them. And it'll build structural connections. It'll also disintegrate them or remove them when that firing pattern gets particularly low. And we'll look at examples of that too. You know? Okay. So we get this idea from Donald Hebb. So it's called a Hebbian synapse because we look at these types of synapses and we call it an assembly of cells or cell assemblies. Okay? And the idea is that it could form the basis for how memories are sort of stored or formed. Okay? That is how the nervous system can remember things that it experienced before. Okay? So memory is kind of a nebulous term. We introduced this idea last class, and instead we can consider it a change in some type of responsivity based upon an experience that happened earlier. Okay, so here if you might say that they're storing memory traces, that is, they might be responding differently after a particular experience. Okay, and we can infer from that it seems that something is remembered. Right? And we talked about that in the opposite sense, when after experience nothing happens. Like when you whoop your kid's rear for getting suspended in school, and after that stimuli, the kid goes back to school and does the same mistake or whatever it was he got in trouble for, and you say, why didn't you learn? We were looking for a change in behavior or a change in something based on that stimuli. So we consider memory to be that function, and we'll look at that in the context of synapses. Okay? Now when we measure synapses, and when we measure cells in synapses, you guys are all really familiar with the idea of graded potentials now, right? Really familiar with the idea of postsynaptic potentials, EPSPs, IPSPs, and the idea is that you can take a needle, stick it into the cell, and you can measure its electrical voltage, right? And you can measure how that voltage changes as a function of the input it's receiving from cells, giving it signals, okay? So what you can see is that this might look like our normal PSP, right? But then what you do is you give it a certain stimuli, all right? So something you might call, it's called tetanus, high-frequency stimulation. Basically, you electrically zap the ever-living heck out of it. Just a ton of a bolt of electricity, just jolt that sucker, okay? Well, then you go and you measure the postsynaptic response to that cell again afterwards, okay? So you started off with a PSP that looked like this, our normal PSP. Then something happens, you go zap the heck out of it, give it a bunch of electrical stimulation. Well, afterwards, after that 100 hertz stimulation, look at the size of the PSP. Got bigger, right? Now, neurons don't have consciousness, so far as we know, but you might say that the cell learned. It learned something about that stimulus that's going to respond to differently. Make sense? Okay. Same idea here. You can take it in the opposite direction. You give a really low, boring frequency. Really low frequency. Okay. It might have started off with a high postsynaptic response, but kind of like our Taylor Swift synapse, where it kind of retracts and goes away. After a really low frequency, this is only 1 hertz, we're going to see that this is our normal rate. Okay, this is the higher one, and it gets even smaller than the normal. So you might say that this is potentiated, which means it's higher, right? And down here, it's depressed, right? Because it gets lower. Now we're going to look at how those changes in synaptic plasticity, these changes in these cell assemblies, are evident to persist in a long-term fashion over time. So what we'll see is it's called long-term potentiation and long-term depression. Make sense? We good? Okay. So this is one way to think about it. Okay. These Hebbian rules. Cells that wire together or fire together, wire together. Okay. So let's say you have a simple 3-cell system here. Okay. Now, the colors are useful. Okay, because it shows strength and weakness. So here you have a weak input. So it's basically this light pink. Okay? Here you have a really strong synapse, shown in deep dark red. So the magnitude of the color tells you really useful information to understanding this figure. Okay? Now, there's really no strength to this synapse, but there's weak strength to this one. So because it's weak, from this weak pink color, there's only a little bit of weak pink that comes in to this cell. So it's only moderately affected. Maybe it's not even reaching threshold to fire. You guys follow? Okay. Now, let's take a look at if there's stimulation instead. So this is still the same. This is weak and this is weak. Okay? So when we started, is the weak synapse enough to really generate much action over here? No. Okay. Now, let's say this is still just as weak, but it's paired together, okay, with a different synapse that's strong. So this fires at the same time that this fires, okay? And so what it does is it induces a big change, and this now becomes very red, showing it's very depolarized and it's firing too, okay? So now you have a weak synapse paired with a strong one, okay? Or associated with it. Talk about associative LTP. Okay? And what you can see then, what results after that, is what used to be a weak synapse over here is now a strong one. Because it went from light pink to dark red. And now, whereas it started off not being able to do much for this cell, now it's ringing its bell all day. See what I'm saying? Does that make sense?
I have a question.
Yes.
So, in the third picture, does that mean the strong synapse changed as well? Because it's not showing red on the strong synapse.
So, good question. Does it mean that it's changed as well? What it shows here is that this cell is not firing in this particular situation. Okay? It shows that when this cell does fire independently on its own, it has been strengthened from its initial point because of its pairing together with a stronger input. It's basically like the movie Hitch. Have you guys seen Hitch?
Yeah.
With Will Smith and Kevin Smith?
So cute.
Yeah, Kevin Smith is trying to dance. He's making the pizza. Make the pizza. Yeah. Will Smith is like, no. Don't need no pizza. They got food there. Right? You dance like this with your... That's how he said dance, right? Not all that's... Okay? Why did I say that's like the movie? This is like Kevin James's character. The hapless romantic. Right? Can't really get it right. Very weak stimuli. He's trying to stimulate his lady friend over here. And she ain't having it. Not even paying attention to him. Just a little bit, but she's like, nah. Call me maybe later. Right? Yeah. Well, what happens? You get a strong connection coming here. He hired Will Smith to come in and help him, right? Show me how to get this right.
Yeah.
Right? So now they pair together. And they both basically start working together to sort of stimulate interest from the third party. In this case, the lady in the book.
Allegra Cole.
Yeah. Allegra Cole was the character. Yeah. Okay. So, how did it work in the movie? The whole reason that he hired this date doctor, Will Smith, was so that later on, Will Smith can go away and he will be independently strong of a connection. It is. I just thought about it right now, too. I'm pretty pumped about this. That's good.
Well, for those who've seen the movie.
Yeah.
I'm like,
That was much better.
yeah.
Okay. So, these are some. Yes,
So
ma'am.
that question would be, is how is long-term potentiation like the date doctor? Is that how that goes?
Yeah, that could be a question. You could think of it in that way. You might think of it, I don't know, off the top of my head, you might think of a question such that it's not just LTP, but associative LTP.
Okay.
Right? Okay. So we see this framework now.
Is
Yes,
there a
ma'am.
question? Just to connect the idea, you need associative LTP to reach long-term potentiation?
In this is one way that you can see how associative long-term potentiation can help to strengthen other synapses, to make cell assemblies. We're going to break it down into just one cell to another cell. But this is about seeing that application that applied to how you can build a broader network.
Okay.
So again, when we came back here, we talked about cell assemblies. You're building assemblies of cells together.
Oh, okay.
Make sense?
Yeah.
So it's because we will dial into this one cell to one cell.
Okay.
But then the kind of broader question that I kind of remains unasked oftentimes is like, well, whoop-de-frickin-do, one cell to one cell. How does this build in to make these cell assemblies, to make it a bigger effect? And this is the conceptual framework that helps to understand that. So, okay, so we'll talk about these basic idea with LTP. It's thought to form the cellular basis for how we might be storing and retrieving our memories. And it forms a really nice way to consider a cellular basis for classical conditioning, Pavlovian conditioning. Pavlov's dogs, yeah, you're associating one stimulus with another. Well, that's basically what we're seeing in our cellular model here, the association of one stimuli with another. Okay? We already talked about the idea that LTP is basically fostered by high-frequency stimulation versus LTD fostered by low-frequency stimulation, that depressive effect, weakening of the synapse. So one good example to think about for that, when I was in graduate school, one of my buddies was, we used to work out together a lot. He was a really intense athlete. He was like a Marine Special Forces guy, and he was getting his PhD in neuroscience once he got out of the service. And we used to work out, hang out. It was a good old time. And then I didn't see him for a couple years. I went to postdoc somewhere, and we caught up again in a meeting. And I said, hey, how's it going, blah, blah, blah. And he's like, yeah, I haven't been able to work out much, this or that. I haven't really been able to get much time. And when I do, it's just this slow-moving job. And it's not really doing it for me. And he was explaining how it wasn't getting the same intensity that he was used to. And it made him worse, actually. And I just looked at him, I was like, yeah, it's LTD versus LTP. And he was like, yeah, that makes a lot of sense. And they're basically, you know, when you have very low frequency stimulation, just kind of a slow jog, you're never going to really grow and get bigger and faster. Right? You're gonna grow your muscles, you're gonna grow your cardiovascular endurance when you push high frequency stimulation. Will help to foster that growth. And just conceptual level. So let's see here. So when we talk about these things, we'll look at a particular model of LTP and look at this increased synaptic strength. Okay? And the way that it's typically done, it was first basically discovered by two guys named Bliss and Lomo. Okay? And what you do is you use a basic setup from hippocampus cells, okay? And you can grab one cell that's making its axonal projection to a second cell. Now you record the postsynaptic response, just like we looked at a few slides ago, and you stimulate with that electrical, you jolt it, right? Okay, so when you do that, do you remember that, I have a marker, but do you remember that when we looked at that postsynaptic response and then how it got potentiated and grew bigger? Just a few slides ago, those red lines? That difference, you can subtract. You measure what's the amplitude of one versus the amplitude of the other, and you subtract it. That's your growth, right? Okay, so you take the amplitude that you measure here, okay, before you stimulate, okay, and that postsynaptic response, that sort of difference, it's basically at 0. Then you give a ton of depolarizing pulses with EPSPs. You give this ton of electrical stimulation. Jolt the ever-living heck out of it. Okay? That's during this brief period of time. Look at how big that response that you measure, right? That's what these dots are. It's the postsynaptic potential amplitude. How tall of a graph do we see? That's what this is saying. It gets potentiated way higher. Okay? And does it go back down? Does it go back down over time? Because this is time now. It goes down a little bit. But does it go back to its baseline?
No.
No. Because this is the baseline back here. This is stimulus. Okay? And afterwards, everywhere that over time, over the long term, everywhere that you can see that it maintains higher levels than its baseline is considered the long-term potentiation, LTP. Based on this basic modeling framework, okay? Just as a snapshot, this is a coronal slice of the hippocampus. So we talked about getting this prep from these two hippocampus cells. This is where it's coming from. Okay, so this is a little slice through a rodent's brain. Okay, this is a hippocampus. Now we'll talk about the hippocampus in a little bit more depth as we move along, and certainly next course in the broader level of anatomy. But the idea here is you have information coming in. It makes what's called a trisynaptic circuit. Tri meaning 3, so 3 synapses. Okay? You have it coming in through a pathway called the perforant pathway. It's going to make its first synapse, okay, onto an area called, in the hippocampus called the dentate gyrus, or DG. Okay? So the dentate gyrus has cells that are called granule cells. Because they're very granular, they're very tiny, and you can see them and whatever. So it's going to make an excitatory connection, that's what the little plus sign is, on our first synapse of the trisynaptic circuit. Okay? So you have information coming in from the perforant pathway, synapses on granule cells in the dentate gyrus, and those granule cells are going to send their axons over to another cell. Now, those axons are called mossy fibers. Okay? It's just an axon. Okay? So cell 1, or synapse 1, sends a projection to the second synapse. These are also cells in the hippocampus in an area called CA3. Now, we've talked about pyramidal cells a lot in the class. That's just the type of cells that these are. So CA3 is going to receive input from the granule cell. Okay? These pyramidal cells are going to send that information to the third synapse in our trisynaptic circuit, which is going to be over here in CA1. Okay? They're also pyramidal cells. Now, just for reference, as much as these were mossy fibers, these are called Schaefer collaterals. So that's just a sort of an anatomical orientation in case you see these terms being used. Okay? And all we're doing is we're measuring LTP from this cell to this one. Okay? So the CA3 cell to the CA1 cell. And that's it. So that's the basic model. And like we talked about, we can see that increase after stimulus in our postsynaptic potentials. Make sense? Yeah? Pretty cool, right? Okay. Now, just for good measure, any good scientist, we want to show, in order to know something, we should also be able to falsify it, right? So how do we know that that's not just, how do we know that's not crap in the sense that Well, that's just how all hippocampus cells. It doesn't matter if you stimulate them. Just they're always going to grow in that postsynaptic response. Well, what you can do is you can measure that cell's response to one cell that does get stimulated and another cell which does not. And that's all this is showing, is we see it in the cell that was stimulated, but this one, okay, was not stimulated, and so it doesn't show that difference. Does that make sense? All right, we keeping it reasonable here? Okay, cool. So that's the idea. And this is just showing this a different way of plotting that data. Okay, so these are our postsynaptic potentials. We saw that there was LTP here, and there's not on the pathway that was not stimulated. And when we look in this graph here, this is the one that was stimulated, this is not. Yeah?
So the non-stimulated ones do go back to the baseline. Is that over time, eventually they will. And then it's gonna.
Well, the non-stimulated ones never leave baseline. That's what the yellow is.
Why are some of them higher though?
Oh, this? Oh, you mean like here?
Yeah.
Here? Oh, that's just normal biology.
Change out.
Yeah, so, I mean, you've probably heard me say this a couple times in class, but it's a pretty good maxim to remember. Biology is messy. Like, we like to think of, like, you know, science as this, like, sort of engineering thing where things are just always pictured. No, biology is really messy. Nature has a lot of variance. There's a lot of heterogeneity. That's normal. It's just, you know, stuff is a little different. Stuff's popping off, you know.
A little bit better to say it just stays around baseline.
Yeah, and when you take statistics classes, you'll get the ideas dialed in of not that it stays around baseline, but that it doesn't have any significant change from basically chance. Remember I said it's messy? This is stochastic, just random variability? Chance. What we want to know is if you stimulate the heck out of a different neuron, will it show a change that happens to be reliably different than chance probabilities? Or random probabilities. And that's exactly what this is. So it's a really nice integration of your stats class concepts.
Yeah, I've done those.
And actual, what's that?
That helped me understand because I've done those.
Yeah, totally. And so that's why we do the stats. Especially why we do the stats, because we know that biology, psychology, it is super messy. And we don't know for sure if what we just observed in our microscope, in our laboratory, in our observatory, was just dumb luck chance. Yeah? Okay, cool. So we got all that kind of good stuff. We can see that it can be evident in not just slices, but also freely behaving animals. It can last for weeks or more, and it seems to have all the hallmarks that we know of what models of memory ought to exhibit cellularly. Now, the way that this happens, okay, just going to get into this kind of briefly, but the idea is that you can have our presynaptic terminal and our postsynaptic one, okay? You have basically glutamate is the neurotransmitter used in these hippocampal cells, okay? So glutamate's going to be released, and it's going to operate on the postsynaptic cell, and particularly, it's going to, well, remember we said that there was different kinds of receptors for glutamate? Yeah. We talked about it in the neurotransmitter lecture. We also talked about it in excruciating detail on the vision lectures in the bipolar cells. Remember? We talked about there being AMPA receptors and NMDA receptors. Okay, so AMPA receptors are going to respond first. Why? Huh? Why is AMPA receptors going to respond first to glutamate? Well, they're ionotropic, right?
Yeah.
Okay, so that's easy enough. What's going to happen, you're going to have that depolarization by sodium, okay, and what's going to happen is our NMDA receptors don't care. They don't care that there's all this neurotransmitter out here, even though neurotransmitter is binding to the NMDA receptor. Why doesn't it care? By caring, why doesn't it respond? Why doesn't it open its channel to allow the goodness to flow? Well, it turns out it's like a cork in a wine bottle, stopping it up. Okay? Now the cork, in this case, is a magnesium ion. The magnesium ion lives in the middle of this pore. And it blocks anything from coming in and out. Now, magnesium is a positively charged ion. And so, if you want to move it, it's very difficult to move in a mechanical way. Remember we talked about mechanical receptors, or mechanically gated channels? We also talked that you can get stuff to move, though, through electrostatic forces. So what happens is the positively charged ions of sodium, if enough is released in that tetanus, in that high-frequency stimulation, enough of that sodium comes in through the AMPA receptors, becomes positively enough charged that it forces the NMDA receptor out of just being lodged in the middle of the receptor, dislodges the magnesium blocker, and now it's open to allow calcium to come in. Now remember, calcium is like ready to roll. It's like, all right, you guys have a nice little soiree going with sodium coming into the party. No, we're going to make this a jam. And so when calcium comes in, because the concentration gradient is so big, it just rushes in there. And calcium is going to be the player that then will make that cell respond later too. Remember, through synaptotagmin and all that kind of good jazz. So calcium is a big player, and that's why it plays such a big role, or the way that it does, by virtue of dislodging the magnesium receptor. Okay? So when that happens, okay, our information is coming in through the AMPA receptors, now shown on the right-hand side. Okay? So just, so you're not too confused. Here, our AMPA is on the left side, magnesium is on the right. Same information, just switched around. Here's your AMPA receptor, glutamate binds to it, sodium comes in, induces a positive change that will basically move that magnesium out, allow calcium to come in. Okay? It's going to activate something called CAM kinase 2. Okay? It's going to induce a bunch of stuff. You don't have to know all these details down here. Okay? Just know that all those biochemical reactions happen. And it'll send a feedback loop back through retrograde signalers, like gaseous nitric oxide. Yeah, you guys remember that from the first test? Okay? Which will tell this cell to release more. Keep the party going. Right? It's like we used to have parties when I was in school, and it's like, maybe you thought you were going to have a pretty good jam. Like, you order a keg, it's a good time. Well, word gets out, everybody starts coming over. It's a real big party. Everybody comes in. You got to call out for more beer. Yo, somebody bring another keg. You got to bring more. Okay, fine. It's a positive feedback loop. Now the party gets really big, right? Okay. So that's kind of the idea there. Okay. And what can happen is when it activates that CAM kinase, it can then result in making more. These are little AMPA receptors in vesicles, and it will insert it into the membrane. So, why, upon a second stimulation, do we see LTP? Why do we see that responsiveness elevated after you have that high-frequency stimulation? In the same cell. Why will the same cell give a bigger response after the initial stimulation than it did beforehand? It's not because of calcium. I'm sorry?
Both ion channels are open now?
It is not because both ion channels are open. Think about the question I asked. It was why. So those are all answers for why, or excuse me, those are all good answers for what happened during the high-frequency stimulation. High-frequency stimulation induced positive charge coming in. It induced kicking out the magnesium blocker, which induced more positive charges rushing in particular. It induced calcium to come in, bind with CAM kinase. But I asked, why does it respond differently after that?
It's resisting more glutamate.
It is not because it's releasing more glutamate. So this is releasing more glutamate, but this cell is not responding any differently necessarily. Yet, let me explain what the answer is. Okay? Yeah, just all right. What happens, it responds more. You see that, you can measure that bigger response that we've looked at now for 20 minutes because it adds more receptors into the membrane to detect AMPA, the ones that are the first responders, you get more of them answering the call. Does that make sense? It's pretty cool, right? So it'll look like this. You start with these two suckers, AMPA and NMDA. Yeah, okay. It's activated. Positive charge is in, kicks out magnesium, calcium comes in, activates CAM kinase. CAM kinase is going to activate this extra AMPA receptor to be inserted into the membrane. So now, we talked about this in the context, there's a party happening, now it becomes a jam, right? Well, you've got all these people outside, you got a jam now, not just a party. You got to open up more doors to get them in. Now you've added a second door. More people can come in and out. Is that a helpful analogy? Okay. Pretty cool, right? Yeah.
So for as long as that's happening, are they constantly like regenerating or making new receptors?
Is there a point where it ever rests?
As long as it continues to be stimulated, or?
Yeah, I mean, there is a point at which it'll like saturate. I mean,
Strong can make it.
Yeah. Or does it recycle like often? Sort of thing.
well, yeah. So it will recycle too. So if it's not, so again, when we talked about motor systems, we talked about somatosensory systems, neuroplasticity, right? Use it or lose it.
Yeah.
In the structural senses, right? Here we're seeing that same principle evident at the synaptic level. So when you're using it, you're adding more receptors in the membrane. But let's say, to answer your question, that you don't use it anymore. For whatever reason, it starts to go away. Yes, they will be retracted and endocytosed back in. For good reason. Now, can you overdo it? Maybe. But cells are never active all the time. This cell is going to take a break. Because it's going to be exhausted of its own calcium stores. Which is going to not allow it to release any more vesicles for a short period of time.
Does that explain the phenomenon? Like, I've heard a theory that that's why we have like certain time frames where we're at our peak in regards to our memory and how we expunge and exchange information.
Kind of at a conceptual level. You ever just been saturated? I have.
Yeah.
I just told you about when I was saturated. I said, I'm trying and I want to learn this and do well, but it gets to the point to where it overwhelms me and saturates. 2006. You guys are probably in like second grade or something, right? Okay, well that's good. Maybe I'm not as old as I think.
I was in college too, but I didn't.
Okay.
Yeah.
But it'll saturate. We get that point to where like, yo, I can't get more crap into here.
Yeah.
Okay? Then you go on Twitter and then you get tons of crap that still fits. Right? You can always get more crap in here, unfortunately. We just feel saturated. We can't learn is what we're trying to say. Right? Or at least we feel that way. We need a little break. We need a little rest.
Yeah.
Because those cells, right? They're in refractory phase.
Yeah.
Maybe not relative refractory phase. The absolute refractory phase. Yeah? Now, here's a challenge to that beyond it. Here's one reason why we know it works kind of like that too. Because it can't work in the opposite direction. Or when it does, you have some real serious clinical pathologies. So is there a spot you said, well, always just keep making more and more and more? Well, it will, but it will until it stops receiving input. And it has to stop receiving input at one point or another when this has to recharge. Or sleep, that's exactly, which is you're recharging, you're no longer getting stimulation. But imagine it wasn't. Imagine it was just uncontrollable change, just constant electrical activity, constantly firing, constantly firing. Or epilepsy, right? So you have constant activity, that's no good. It's no bueno. Right? Healthy balances, right? Yes, sir.
What's CREG?
Oh, CREB, where is that? Oh, down here. It's just one of the sort of downstream biochemical signaling things. You won't need to know CREB for this class. I don't even bother. Just know it's a player in the game at this point who just don't matter. Okay, so that's kind of the idea there. And when that happens, not only will you add these AMPA receptors, but we will see these to accommodate these receptors, it will grow structurally bigger. So these are electron microscope images, okay? This is a synaptic connection, okay? And these dendritic spines will grow and add, or the spines will, or excuse me, the dendrites will grow and add additional new spines, which is really cool. So we're building up from that molecular level to the structural level here, and it fits with the same concept that we looked about, even the structural changes that are happening in neuroplasticity at the somatosensory cortex, at the motor cortex, after learning to play the guitar better, or after losing a limb, or what have you, okay? And so that's kind of this idea there, where you'll see that second receptor now being able to respond to glutamate more. So it makes that cell more responsive. If you start off with a cell that was responsive here, and you add more glutamatergic receptors like AMPA in the membrane, and you give it the same stimulation, it makes sense that its responsivity is going to be measured higher here, right? That's exactly what we started talking about. I'm glad it makes sense. I hope it does, at least. Does it? Okay. Let's see here. So, come up for air. Any questions? Yes?
Okay, hypothetically, in a patient with like Alzheimer's per se, when you do like cognitive training, does it, how do I word this? Is it like a high chance of success or not based on how fast it's generating like the cell? Is it even like?
Yeah, so in cases of Alzheimer's, it's usually characterized by degradation cells.
Right.
There's cell death happening.
Right.
And any cognitive training is definitely going to be moderated by how fast the cell death is happening, of course. Cognitive training isn't really going to work on, definitely isn't going to replace the dead cells.
Right.
It's just going to foster using and strengthening the ones that are in existence, possibly by these mechanisms.
Right.
Because if you're using it, the more you're using it, you're going to build those electrical patterns. The more those electrical patterns are being built, they're building those synaptic connections. And when those synaptic connections are going to be built, the stronger they're going to respond to subsequent stimuli. So it's practice.
So is that what like puzzles means, like that helps you know some of the
It is one idea what, yes, it's one idea why we think puzzles might help. Yeah. I don't know that anybody's done a direct study looking at actual synaptic measurements of AMPA receptors and crossword puzzles, because I don't know that you could. Because to look at AMPA receptors, you pretty much have to look at like a cell membrane in a petri dish, and it's pretty tough to get that cell to do a crossword puzzle. And you know what I'm saying? Like there's just limitations, right? And this is one thing I wanted to communicate since the first day of class into the last, right? We think about science oftentimes in society. We think of science as a thing, and it's not. It's a process, right? It's a way of trying to resolve information and trying to understand what's true about data and observations that we make. And, and that process is limited. We have to make educated guesses or reasonable assumptions, because we can't connect a synapse with a crossword puzzle.
Right.
Because how do you get that cell to do a crossword?
Unless they create a mechanism or machine that could possibly.
But you see what I'm saying? Yeah. And so all science is we're making good, reasonable guesses. There's nothing wrong with that. Those guesses are based on well-informed views about the available information and evidence. But information and evidence that is available changes, because we're hopefully always collecting new things, because we're exploring and discovering. You know what I'm saying? And so we will get new information and revise our understandings of things. So, like, feels like, we settled the science of X or Y or Z. Like, no, we are exploring. And, obviously, I like to get that point across. So, I'm sorry?
It's not that. That's what you're saying.
No, and just because it's a fact in a book, right? It might be true, but we might learn something new, right? There could be an alternative fact. Okay? So, as they say in the modern day. So, speaking of which, let's talk about some exciting new developments. Some ways that we'll find that these cells operate in the hippocampus. So we've looked at these hippocampus cells a little bit now in the context of long-term potentiation. And we'll talk about this area of the brain called the hippocampus. And the word hippocampus is Greek, and it means seahorse. Okay? So we sort of make our consistent connections with marine biology here. The seahorse, we talked about the giant squid axon and discovering action potentials that Hodgkin and Huxley got the Nobel Prize for. Yeah, you remember that conversation? So, marine biology comes to play here. It's a good excuse to go out and scuba dive. It's for science. You can say that. So, we've talked about these structural changes. We've talked about new synapses forming and how training might lead to integrating a new organization of those synapses. Okay. Now, one application of that is there's cells that were found in the hippocampus to be very, very sensitive to where you are in space. That is, where you are in like a grid. Okay? So Fitcher, you usually sit over here, don't you? Somewhere?
In the other class.
Oh, in the other class, okay. Yeah, see, even my hippocampus ain't working all that great. Right? But imagine that when you're in that seat now, in that place in the grid of this room, you're going to have certain cells firing off. But when you sit in this space over here, those cells will shut up and new cells will start talking. It helps you to get an idea of where you are as we move around in space. We talk spatial navigation, right? So we talked about the vestibular information coming in, telling us where we are in space. That's just sensory. We want to encode that information to learn about it and God help us remember. Yeah?
So is that why teachers say to take a test in the same seat that you normally sit in so you can remember better?
It's related to that, yeah. So sometimes teachers say to take a test in the same seat that you sit in. That's an effect called basically context-dependent learning. The broad concept is similar.
Okay.
I don't think that's necessarily based on place cells per se, but the idea is right on point. And I'll try to get to those cognitive elements maybe next class in a little bit more depth. But the idea here is that you have those place cells. Now, and hippocampus is also known for being very involved in memory for spatial locations and navigations. And one line of evidence that supported that interpretation came from a very popular study known as the London taxicab driver study. Okay, so Eleanor Maguire conducted this study a little while ago, and in London, so London is a very old town, and it's not like our towns that were built sort of in modern era and were planned. It was built over, 1,500 years of one road being on top of another, and then they built this building and it wrapped around, and the roads are basically crazy. Have any of you guys been to London? Rose, you have? Okay, are the roads very neat and orderly?
Oh, they're so bad.
They're horrible.
Yeah.
Tiny. Okay. So, in order to be a taxi cab driver, so I should say, a long time ago, before there was Uber, there were these things called taxis. And the way it worked is you'd stand on the side of the road, you'd have to wave somebody down, and you'd have to ask them for a ride, and they'd come in, the cars were like yellow, they didn't have a mustache on them or anything, and they would give you a ride, and you would pay them money, and they would charge you more money, and people were happy for a little while until Uber came along. Okay? So, in London, those taxi cab drivers would drive people all around, but they had to be licensed, and in order to be licensed, you had to pass a test that showed that you know how to get around the freaking city, because it was complicated, which meant you really had to study the spatial maps, and you had to be very good at it. What they found was basically that people who were taxi cab drivers had extensive spatial memory, and they had much bigger hippocampus volumes than people who were not taxi cab drivers. So basically the idea is that the more you're using this skill of spatial navigation, okay, the more you're kind of growing and strengthening the structure that will even grow in volume. Now, granted, I said grow, but the study was correlational. It wasn't causative. It didn't say it caused it to grow. It said it was correlated with bigger growth than people who did not engage in that task. Okay? And within the taxi cab drivers, their hippocampi were bigger than each other's were if they'd been on the job longer. right? Reasonably good correlational evidence. Not causative. Correlations are okay. We just need to know how to properly understand it. But that was the idea. Okay? And so this is the idea that this structure, the hippocampus, was involved in that spatial navigation. Okay? Now we'll talk about the hippocampus a lot now. Pretty much the rest of the class. It's near and dear to my heart. It's a great structure. It really is. And when we talk about it, I just want to remind you some of the anatomy, orient you to exactly what we're talking about. Okay? So sagittally, we have this sort of C-shaped structure. It's bilateral, by the way. So we see it here on the left-hand side. It'll be going around on the right-hand side, too. And notice you have to take this unique anatomical cut, right? You have to cut it this way and this way to bring out that sort of triangular chunk out to see it wrap around, okay? Now, if you look at it and you slice it this way, you get the coronal slice, and here's our hippocampus in the medial version of our temporal lobe. Yeah? Okay. And, of course, we get it etymologically from the Greek word seahorse. So if any of you guys come down and visit my laboratory at all in the next semesters to either work in the lab or be a research participant and do some memory tests later, I'd love to see you guys. And if you come down, you'll see that since I'm just starting out here, when they gave me the new lab to do, I inherited a lab in the Child Development Center that used to be an infant and toddler lab. So there's all sorts of baby drawings and pictures all on the wall and all sorts of stuff that looks very different from other cognitive neuroscience labs. And there's really cool scene that they painted on the wall in the lab of an underwater like scuba diving scene of these cute little like toddler pictures and characters of little fishies and Nemo and all that kind of stuff. I don't know, like a SpongeBob kind of scene, right? And they put some seahorses in there because that's what you put underwater. It worked out great for me because it's like, sweet, it's like the campus lab. Yeah. So we had them paint over all the kids stuff, and we left that. We left that one undersea mural. So you guys come down and see it sometime. Yeah. So hippocampus. So we talked about that trisynaptic circuit earlier today. That information is coming in. Synapse, one synapse, two synapses, three synapses. Okay. That's our cartoon. If you're an electrical engineer, or you just like to draw it more simply, here's just a bunch of lines, okay? Entorhinal cortex, the information comes in, the dentate gyrus, the CA3, the CA1, and then out, back again, through a part of the hippocampus called the clavicula, okay? So this is kind of opposite to this, okay? So here, you have information coming in, and coming out. You see that? Okay? It's coming in and coming out. It's just saying this is the mirror image of that. It's the flipped. Of course, that's a cartoon, and this is a line drawing. This is reality. This is this is a drawing by Cahal. Remember our good friend Cahal? Father of the selfie? Remember he had that like picture of him doing this? His shirt off?
Homemade cannon.
Yeah, homemade cannon, yeah. He's the original gangster. He's truly OG. Okay? This is what he drew. Same information coming in. Okay? Synapsing on dentate gyrus. Okay? To CA3, CA1, and back after the clavicula. Actually, no, I'm sorry. That comes in. Entorhinal cortex comes in this way. Synapses there. I had my own mirror image reversing around. So, that's the idea here. In the modern era, we can use other stains. Remember we talked about methods earlier in the class? Okay? You can use stains to label cells. And when you use different fluorescent labels, like green fluorescent protein or yellow fluorescent protein or blue fluorescent protein, etc., you can tag all these different cells in the hippocampus. Okay? So here's that hippocampus again, and there's these different cells and their projections. Okay? When they came up with this, they called it the rainbow mouse. It's a stain from a mouse's hippocampus. So rainbow. That's the colors. That's all they got. That's it. It's pretty cool, right? Okay, so let's talk more about these place cells that we introduced. So we're talking about them in the hippocampus. Let's say you got to navigate this whole map and all this kind of stuff, and they will have electrodes dialed into different parts of the hippocampus, not just different parts, different cells. These different cells in the hippocampus, they all have different electrodes dropped into them in this experiment. Okay, and as the mouse goes and runs in a maze in different places, different colors show where different cells started firing. Notice that when one color goes on, the other colors go off. Okay, so you're showing some cells like one thing but hate the other. Right? Then you go to a different place, and those cells respond differently. Okay, so you're dissociating their responsivity to different places in three-dimensional space. Okay, for learning, for encoding that information, because they fire off when you're back in that space. Okay. And they will remap when you're placed in a new environment, okay? So it's, again, this is the learning and memory phase of that sensory information. Now, when we do that, we can see a couple things, okay? First, this discovery that cells work like that, okay, was recently recognized in 2014 with the Nobel Prize. Wow. Okay? It's pretty cool, okay? I went to a team of researchers. Actually, there are different teams. Okay, this is John O'Keefe. He was the guy who discovered those cells. All right, discovered them in a paper in 1971, and it was published in kind of a small-tier journal. It's actually a pretty lowly ranked journal. And you know what happened then after he published it for the next couple decades?
No.
A lot of people did not believe it. Nothing was almost what happened. Now he cranked out study after study after study, but for a long time, he had very, very difficult time convincing people that that's how they worked. And in fact, that they were doing anything like that at all. And there's still some people who argue that they're more involved for processing information like timing. So Howard Eichenbaum at Boston University has a recent paper suggesting that a lot of these cells' job is to do relational binding of temporal information rather than as much place coding. But the idea began to pick up steam over the decades. And one of the things that made it much more conclusive findings to support his came from a team of researchers in Norway, the husband and wife team, Edvard and Britt-May Moser, May-Britt Moser. And they found similar pieces of information, but in the entorhinal cortex. Okay, so we come back here, we have the entorhinal cortex, the EC, okay? EC coming in. So this is our hippocampus, and this is the entorhinal cortex feeding into it. They found that if you could find similar types of cells here as here. So these are doing place, okay, and these are more grid cells. So you kind of want grid driven. So they're going to map together to give you your sort of GPS coordinates of the brain, okay. And so when those things came together, those models, they had a paper I think in 2004, I want to say, on grid cells. The year might be off a little bit. And I'd be remiss if I didn't say the role that Southern California played in these series of discoveries, because at UCLA, there was a very, very important breakthrough that came through in the lab of, what was his name? Itzhak Fried. He had a researcher in his lab, Arne Ekstrom, who was the first person to identify place cells in human beings. Because for the while, John O'Keefe had a tough time convincing people of any of this. Finally, people are like, all right, fine, all right, whatever, they're in mice. Right? But then what all the haters at the bars tend to say then, once you finally got them to agree, they're like, all right, fine, you're right, we already knew that. We knew that the whole time. Right? But they're like, whatever, it's just mice. It doesn't mean anything, because all we care about are humans. It's not like, there's nothing reason to think that human brains work like that. And Arne Ekstrom conducted that study and actually found human place cells. How the heck would you do that? No, really, how would you do it?
On a person, you're talking about?
Yeah.
Maybe a person?
A live
You
person.
have to be alive because you need to see the actual activity, right?
Yeah, it's pretty tough, right? But EEG's at the scalp, right?
Functional MRI.
Functional can't get down to the cellular resolution. You have a lot of these limitations.
Like brain surgery?
Brain surgery, you said that. Oh yeah, good call, Rose. Brain surgery, yeah, we'll talk about that, okay? So, in rodents, when you were able to do it, first of all, you could see that you can first start off with a water maze. So you drop a mouse into a big pool, okay? It's like one of those big above ground swimming pools, like a kid's pool or something like that. And you hide a platform in there that is clear plastic color. Now you drop a rat in the water, and it's going to act a lot of the ways that you would act if I went and dropped you in the pool down the way here on campus. You'd shriek and go, ah, what are you? Don't throw me in the pool, right? At least you probably would. And then you try to climb out, right? So the rat tries to, they swim all around, they're trying. Now eventually, okay, they learn that there's a platform there, and when you drop them in, after a couple trials, they don't do any of this mess. They just go, yeah, I know where I'm going. And they have a little island. They don't have to swim. They're like, sweet. And hang out here and have a Mai Tai. It's like one of those bars on spring break where they have the bar built into the pool. You guys ever been to that? Yeah, it's really awesome. It's really cool. You just swim right up. You can drink. You can drink and pee at the same time.
Someone's doing it.
Everyone's doing
Come on now.
How do you pay for your drink?
It's all inclusive, but a lot of us.
Yeah.
Okay, that's
Or you can have plastic too. You can bring your credit card and don't care about the pool. But if you lesion the hippocampus, you cut the hippocampus out of the rat, they're going to look for it, they're going to find it, but even after 10 trials where they're used to going straight to the bar, right, they're still looking all around the pool. Because they can't remember. So again, implicating the role in the hippocampus for this spatial navigation. Sorry? I've never seen Finding Dory.
All these movies that you quote, you've never seen Finding Dory?
Look, here's my scope of movies. Right up until about the time I had to start studying seriously in school, in grad school, and then I dropped off. That's why I'm stuck at like the Shrek stage. Okay? Like right about that era, like I couldn't watch any more movies, and so all my quotes come from about then, and whatever happened after that, Dory or Nemo or whoever, I got nothing for you. I'm like these. Yeah. So, anyways, let's see that when you then record and say like a circle maze, it's not really a maze here, it's just a pathway. But again, we see those patterns of firing are going to be different in different cells. That's what the colors are. Colors are electrical activity patterns from different cells. Okay? And we saw that in this S phase. Okay? And we talked about how these grid cells can build up. Okay? So they're going to fire when you're in a certain pattern or a grid. Okay? As you go around and you hit these little points on the grid, okay, you have the place cells here in orange. Okay, they're just going to fire right when you're in this place. Okay, but grid cells in blue. Okay, so this is hippocampus, entorhinal cortex. Right? The hippocampus place cells were discovered by John O'Keefe. Entorhinal cells, for the grid cells, by the Mosers in Norway. Now, I should say, really, really kind of little cool anecdote. Just this past November, I was at in San Diego. They have an annual meeting with 30,000 neuroscientists, and it's a bunch of it's basically a big science nerd convention, presenting posters. Like in 7th grade, you know, like your little science fair poster. It's basically that for grown-ups. It's really awesome. It's really cool, and usually it's a bunch of grad students and postdocs presenting their posters while all the old professors schmooze and hobnob or whatnot. And when I was there, I actually saw John O'Keefe presenting his own poster, which is really, really cool. I just want to underscore how awesome that is of a guy of his stature to be there presenting a poster on the floor, just chatting with people. It'd be like if you went to the gym to work out and like Michael Jordan's just in there like shooting free throws, you know, just kind of doing his thing and just playing pickup games with you. It's freaking awesome. So John O'Keefe was really, really cool. We got to talk to him, and you know the thing is, nobody expected it, so nobody was there. He was just there by himself, just poking around like the other grad students, just kind of standing by his poster alone. And I was like, walking by. So you guys go talking. It's really, really cool. Yeah. So, and this is Arne Ekström with the human play cell. So what he did is he did virtual navigation. Virtual navigation with like a video game in VR. So you had a little joystick and you moved around while you're undergoing neurosurgery for epilepsy. So before you cut out the area that's bad for epilepsy, you do pre-surgical mapping. We talk about this a lot in class, right? And so they're able to have an electrode down in the hippocampus. When you're moving around in three-dimensional space in the navigational game, because you can't like having to go walk around in a taxi cab in LA, right? He was able to identify place cell activity. It's really, really awesome. So these are the big three sort of papers and players there: John O'Keefe, the Mosers, and Arne Ekstrom. And Arne is a really cool guy. I had a chance to work with him in grad school a little bit. Super awesome. If you guys are looking for really cool mentors or researchers to work with in graduate school or something, keep that guy on your radar. Really awesome. So we get that, and this is kind of that schematic that they made about when they announced the Nobel Prize. You kind of have your grid cells sort of scaffolding the layer from the entorhinal cortex down to the place cells here that help us to determine your three-dimensional space of where am I and have I been here before. Pretty cool, right? Yeah, I think so. I hope you think so. So we have that, and I mentioned the John O'Keefe thing from last. So we're navigating, right? Let's say you're going through this. Let's say you're here, and you leave this door out through here, and now you've got to get on the bus. Okay? Right? We need to, it's important to have our memory operating, right? We don't want to be amnesic. Right? So you got to know this stuff. And so you leave there, and you start driving down the way, and this guy starts chatting you up on a bus. Okay? And so let's say you're at Neuroscience 2015. It's not in, say, San Diego like it was this past year. Maybe it's, this one is in Chicago. Okay? So now you're there. You're on a shuttle, and you're driving around on the bus, and you've got to take this spatial map, okay? So you're going to have your grid cells going off when you're in different sections of the grid together. You're also going to have your place cells going off when you're in different spots. Yeah? Make sense? Okay, cool. So let's just say then that the gentleman next to you, he tells you that he studies place cells in Norway, and that the field is now much more crowded than when he started, and it's more difficult to publish. What would you say? It does suck, yeah. Well, kind of. So you would tell him that sucks, right?
Try changing your charts.
Try changing your charts?
Yeah, sometimes publishers get picky about it. I'm just being smart.
Yeah, maybe. But what else would you tell him?
Find a different way. Find a new method.
Find a new method. That's one thing, one way to kind of carve out your own place, right? If it's, well, no pun intended. If the field is, yeah. Getting a little busy, difficult to publish, you got to carve out your own spot.
Go to a new destination.
Go to a new destination. That's also a great thing that you could tell the gentleman next to you on the bus. Okay, so perhaps you could, maybe he should join Moser's Lab, like give him a new destination, or you can learn a new method, publish a new thing. Anything else you would tell him?
Team up with other researchers.
Yeah, like maybe he could join Moser's Lab, team up with other researchers. Exactly. And in fact, that's exactly what I told him when I sat next to him. And I took this picture. And why am I here telling you about it? Because that guy is Moser. And I asked him, maybe you should think about joining the Moser Lab. Maybe they'll let you in.
Did he say thank you?
No, what he said is, that is me. Oh, well. Yeah. So we had a conversation for about 10 minutes. He's like, what do you do? I was like, oh, I study the physiology of the hippocampus. He's like, really? I'm like, oh, that's cool. I'm like, what do you study? He's like, oh, I study entorhinal grid cells. I'm like, oh, cool. Those are popular. He made a little joke. I'm like, yeah, I heard they love those in Stockholm because that's where they give the Nobel Prize at. And he kind of chuckled. I made a joke about the Nobel Prize to his Nobel Prize. He kind of chuckled because he obviously realized I had no clue who he was. I knew about him, but I tell the story to you guys, right? We don't just need to know the facts for the test, right? We don't need to just go, oh, what do I need to know for the test? Can you make me an outline? No, you don't just, right? We need to know things beyond the name, right? Integrate the faces, the names, the whole picture. Right? You'll never be tested on a picture of someone's face in this class. But we need to know it anyways if it's not on a test, so you don't end up embarrassed like me. Okay? I say, yeah,
I heard they like the Nobel Prize. They like good cells in Norway these days and stuff. I was like, oh yeah, they do. And she's like, what do you do? Tell me about your work. I said, I study hippocampus physiology and memory. He's like, oh, that's really cool. And I agree with him. I'm like, yeah, you know it is. And I tell him all about how awesome it is. And really, oh yeah, really cutting edge, really cool stuff. And you know, kind of, because I don't recognize him from my field of like human. Like I know those people's faces. I talk with them. So I just figured he must not know the kind of work I do. So I, you know, kind of just elevator talk, you know. And I asked him, like I said, well, maybe, you know, how's it going for you? He's like, well, it's getting a little harder to publish. In the early days, nobody was doing this. I could get stuff out pretty easily. Now my field is so crowded, it's still tough to publish. And I did exactly as you guys said. I took exactly your recommendation. I said, well, maybe you should join someone else's lab. Maybe it'll help you out. And he's just loving it because he's trolling me like you wouldn't believe, right? Just trolling me hard. I'm like, yeah, you should, you know. And I put like my little mentoring hat on. I'm like, you can help this guy out. Yeah, give, yeah, give, you know, give a little pep talk, you know, like.
What was the lesson you learned?
Well, to learn more than what you just need to know for the test.
I think there's more.
So yeah, and I said, well, you should do that. He said, oh, that is me. And I said, oh my God, I know who you are. Like, I'm sorry. It was actually dark.
Yeah, there you go. It was dark. I couldn't see.
Yeah, like so the camera flash went off in this. It wasn't this light. This was just the flash from the camera. And it was, you know, 15 to 12 hours in the conference. It was a long day. And you just never expect that this guy's going to be taking the bus next year.
But didn't you think it was cool that he didn't like put you down or anything like that? Like he was just like humble about it. He could have been like, dude, what the hell is wrong with you? Like I am, but he didn't.
No, it was awesome. Like, and he was humble enough to not like take a limousine back to the hotel. He's sitting there on the bus with the rest of us schmucks. And I was so impressed by it. And he actually really loved it. And so he starts laughing at my face. Now, granted, I'm, you know, chatty Cathy 10 hours into the conference on the bus, right? And I'm just talking. So the people around me, they all hear this go down, and they're all just laughing at me now. Because they must, the whole time they must have been like, yo, that guy's next to Moser. And now I'm just going off and going
That is awesome.
And now they're laughing at me, they're standing up like, you know, and anyway, so what he said after he said, yeah, that's me. I'm like, dude, I'm so embarrassed. I said, I know about places, I know about your stuff. I work with Arne Ekstrom. We publish papers together because we had. And he's like, oh yeah, I know Arne. We were at Davis together. Blah 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 blah. I'm like, no, you gotta believe me. Like, yeah, really. I'm telling you, like he rolls with my PhD supervisor. Like they're all like tight, and I'm like, no, really. I'm like name dropping like crazy at this point. and he's like, no, it's okay. He's like, I actually really enjoyed not being recognized. Like, this is really cool. I just like having like a normal science chat with people. He's like, these days it's hard to have that normal conversation. Everybody just wants to take a selfie with me. And I'm like, well, you know, now I kind of have to.
Now that you mentioned the selfie.
Yeah, right? So, yeah, I mean, that was them, and that was our selfie. You know, and then
was photobombing.
Yeah, she was definitely photobombing me back. And because I got a chance to pitch him about my research, I looked at the analytics on my research presentation I gave that day about my hippocampus work on YouTube, the analytics. One view from Norway.
He looked at it.
It's pretty cool. So, the point of the story is to remember stuff. We're having lecture about memory. Don't just learn stuff to rote regurgitate on a test. I know it worked like rock stars in high school, but like you're gonna have to know other stuff beyond the test, especially in life. So please use that as an example to tie into the lecture on memory. Yeah, that's the idea with the hippocampus and memory. In other animals, okay, it will grow, or basically there's a correlational or corresponding size in the volume of the hippocampus based, again, on how long that taxi driver had spent doing spatial navigation. Okay, so this is just the data from that taxi cab driver thing or study. Let me see what else we got here. We talked about dendritic spines earlier for the LTP conversation. This is just some more data on that. and you asked about Krebs earlier, but I said don't have to know Krebs. Remember we said that you get those AMPA receptors increased in the membrane, inserted by virtue of CAM kinase. You guys remember that? Okay, so if you tag CAM kinase and you say every time CAM kinase is active, fluoresce like a mofo. Just fluoresce like crazy. You put a tag in there, and you can tag CAM kinase. That's what this is showing. Normally, here's your dendrite, okay? And there's a little spine connecting them, okay? Okay, when you stimulate, you tag it for CAM kinase, you see that activity increase. And afterwards, you see it persist, okay? And this is that increase. This increase here, this higher elevation, is that red dot. Just showing that role that CAM kinase plays. And let's see what else we can do. Same idea here. We see increase. See this big old honking dendritic spine there. Much bigger and expanded than it was before LTP. Okay. So structural changes happening as by account of long-term, or excuse me, synaptic plasticity. Let's see what else we got here. We've talked about these details. Oh, you know what it is? This is exactly. We talked about the long-term depression earlier. That's what this is. This is the downside. So we've talked about long-term potentiation. This is a D, for those of y'all who can't see it. Yep. And the way that that works, surprise, surprise, you suck those AMPA receptors out of the membrane by virtue of the low-frequency stimulation. Okay. So it's just easy way to do it. It's just the opposite way. Okay. And so this is just sort of summarizing where we've been. A couple features about LTP. It depends on the state that it's in. Okay, so it has to have AMPA receptors and there has to be depolarization to take that magnesium blocker out of the channel. So in that case, it works as it has to detect a coincidence of both of those things happening. There has to be a positive depolarization sufficient in both the AMPA receptors and the NMDA receptors. Okay? It has specificity. It's only going to work at those sites that are concurrently active, but it also has associativity, and that a certain weak stimulation can produce LTP if the inputs around it are also strong enough to depolarize it. That's what we started our conversation with that little pink blob making that big red blob showing the depolarization of the cells. You guys remember those weak and strong synapses? So what we tried to do is start with that very simplistic overview of a pink color and a red color, and now we've dialed it into the molecules, to the proteins, to the cells, and built those molecules and protein channels into the cells that form the networks in the hippocampus, and how those networks interact between grid and place cells to give us our larger cognitive experience of where we are in three-dimensional space. Help us to navigate our spatial directions. It's a lot, isn't it? It's a lot of stuff you guys just journeyed through. Yeah, you know, I mean, that's really cool stuff. I'll skip through some of these things. And that's kind of the idea. When we pick up in the next class, we'll talk about amnesia patients, okay? We'll talk about other cognitive elements, okay? I posted an article yesterday on Patient HM. It's an easy article to read. It's from a magazine in 2010, Esquire magazine, so it's an easy read. Zip through it. It'll give you some background. It's written by the grandson of this famous surgeon who chopped out both medial temporal lobes of a human who became densely amnesic and the most famous brain damage patient in modern history. And we will talk a lot about that person, patient HM, next class.
Made me cry.
Sorry?
Made me cry. It's like his
Yeah,
stepmom.
It's a story of humanity.
One doctor,
Yeah.
1 out of 7 doctors said he was schizophrenic. So.
Yeah. So we'll do that. I think we got what, 1 minute left. Okay. So are you guys feeling a little bit better about your grades right now? A little bit more stabilized, but room to grow, right? Biggest room in the house is room for improvement. We still got a really good chance for that on the final. Okay. If anybody wants to work on an extra credit project, come see me after class. It'll take a couple hours of your time, but it'll involve reviewing some lectures and turning them into videos. So it's an optional project idea, and depending on how many people want to do it, come and talk to me, and we'll either set you up with it or set you up with Teams. Okay? I will see you guys, hopefully downstairs, whooping my face with pie, and otherwise have a wonderful week, and I will see you on Thursday. Yes. That's okay. No, it'll take a couple hours to do the project. Yeah.
Oh, okay.
Are you interested in it?
Yeah, I'll
Okay. Yeah, hang out. If you guys are interested, hang out for a bit. Maybe doing video editing?